I'm Dr. Neeraj Khatri Sapkota, and today we are going to again continue with the same topic regarding with the blood and its effect on the body system when there is hypoxia. So hypoxia will be relevant with the more formation of RBC. The process is known as erythropoiesis, which gets stimulated by the hypoxia. And for the synthesis of the RBC, we need one of the very important hormone which I have told you in previous video as well, and that is none other than erythropoietin. So this erythropoietin hormones is going to work on the formation of RBC through the erythropoiesis process, and this is affected by one of the factors known as hypoxic inducible factor. And this hypoxic inducible factor is an intracellular signaling molecules that accumulate as a result increasing the synthesis and release of erythropoietin. So hypoxic inducible factor is going to stimulate or increase the release of erythropoietin from fibroblast cell of kidney and liver. So what will happen, the resultant is that this condition may be caused by iron deficiency in which there is insufficient hemoglobin in each RBC, high altitude, or pneumonia. Three relevant conditions where there is secretion of erythropoietin includes iron deficiency condition where there is not enough hemoglobin in RBC, for example, the condition where there is insufficient oxygen in the environment, such as high altitude, or in condition where there is less diffusing of the oxygen from the lungs into the blood, so that there will be less oxygen in the blood cells, and the condition one of the condition is pneumonia, so which cause reduced availability of oxygen. And this reduced availability of oxygen will cause the formation of hypoxia inducible factor, which is a internal intracellular signaling molecules, and that stimulates, let's say if this is hypoxia, if there is less oxygen, then there will be HIF, and this HIF is going to work on the kidney fibroblast cells to produce more erythropoietin hormones, and this erythropoietin hormones is going to work in the bone marrow. So let's say if this is erythropoietin hormone, then it is going to work in the bone marrow, and in the bone marrow the hemocytoblast cells or some of the progenitor cells or hematopoietic stem cells get stimulated, and the stimulation will result into the increased rate of erythropoiesis. This effect is even stimulated by the testosterone, the male sex hormone, and said to be inhibited by the estrogen, the female sex hormone. Hence, in the male, there is more number of RBC compared to the female. This is what the reasoning is provided in terms of the difference in the male and female RBC count. So you can see in continuation, so erythropoietin stimulates red bone marrow cells that are already committed to forming an erythrocyte, causing them to mature more quickly. As a result of that, this RBC will be formed. Later on, this RBC will be less flexible, more rigid, and more fragile, and hence they are transported and they goes to the spleen or liver, where they will be phagocytosed by the macrophage, and this phagocytosis will destroy the RBC that has already been old, and this will produce the hemoglobin that is present in the RBC. So each RBC cell is very important in terms of it carries iron, and globin proteins. So altogether, hemoglobin contains approximately 65% of the body supply of iron, which is about 4 milligram, 4000 milligram, that is 4 gram. That means in hemoglobin altogether about 4 gram of the iron is present in the hemoglobin. So the liver and spleen store rest of the other iron, while small amount also stored in the bone marrow. So iron is stored inside cells as protein iron complex called ferritin and hemosiderin. That's why this hem, when it is destroyed or degraded, then it will be iron and then it will form biliverdin. So biliverdin will be processed into bilirubin by the liver cells, and globin is further degraded into the amino acid which can be reused for the synthesis of hemoglobin as well. And this iron, it goes to the blood and they will complex with another protein known as transferrin, and this transferrin will just mobilize the iron to reutilize use for the synthesis of the hemoglobin, as the free iron is toxic to the body. That's why this can be concluded with saying that testosterone also induces production of erythropoietin in the kidney. As a result of that, female sex hormone do not do this. So male, this testosterone is male sex hormone. So therefore, testosterone may account somewhat for higher hemoglobin level and RBC count in male. Erythrocyte production may also be heightened by many different chemicals released by leukocytes, platelets, and other condition, some other reticular cells. Therefore, the recycling and processing of this iron and the synthesis of the RBC DNA synthesis and RBC hemoglobin synthesis requires hormones, vitamins such as vitamin B12, and folic acid, and iron, the supplication of which can be done through the ingested food or consumed food that are absorbed from the intestine, and then these nutrients and irons are transported through the bloodstream to the bone marrow, and then that will be utilized for the process of the erythropoiesis. So this is how the life cycle of the RBC is integrated, and then they are formed and they are destroyed. So that whole process takes about 120 days. So 120 days is the life cycle of the RBC, which we generally consider to be the important part of the function of the RBC cells to incorporate oxygen and take the oxygen to the tissue, so that adequate supply of the oxygen to the tissue will take place. Hence the tissue will get oxygen and then it will produce ATP. So that's how our cells are getting survived, as a result of the continuous supplication of the oxygen by the RBC hemoglobin content. So with this, I'm gonna conclude the video for this today's section. So we'll continue another section of the RBC in next video. For now, I'm going to end the session, but before ending the session, if you like my video, then just don't forget to subscribe and comments or like my video. That's what I would request for every audience that see my video. So thank you for concentrating on my video for about 7 to 8 minutes. With this, I would like to conclude the today's video. Thank you.